So it gives me great pleasure to introduce Julian Thayer to you. I've known Julian for decades. I've known him since he was a graduate student. In fact, I served on his dissertation committee many moons ago. So I've known Julian ever since. And there's a lot to say about him, but I'll keep it brief. Julian was a student in the Midwest. He got his undergraduate degree at Indiana University. He then went to NYU to get his PhD. And he is now a distinguished university professor at UC Irvine. Julian also, for those of you who don't know, is a super accomplished jazz musician who performs all over the world. In addition to his musical accolades, he has received many distinguished scientific accolades, including the Distinguished Scientific Contribution Award from the Society for Psychophysiological Research, which is a hard-nosed society and is a very distinguished group of awardees. He also has received the Distinguished Scientist Award from the American Psychosomatic Society. Now, one of the things that really characterizes Julian's work is he has been interested in the connection between our mind and body, broadly defined, and health psychology may be a category that some people put him in, but he is not a typical investigator in these areas. He has brought an understanding of the brain to help us understand the mechanisms through which psychological processes may be expressed in peripheral physiological events, and also how those peripheral physiological events may impact on our psychological states. He is one of the world's leading experts on heart rate variability, which has received a lot of attention, both in the scientific literature and in the popular press and other popular venues, some of which is hype. And I always turn to Julian to, as we say in Wisconsin, to help sift and winnow through the various strands that are out there. So without further ado, let's give Julian a warm welcome. Thank you.
Thanks, Richie, for that kind introduction. Indeed, we go back quite a long way. So I'm really happy to be here. So as Richie mentioned, let's see if we're going in the right direction. There we go. Let's see. There we go. So, as Richie mentioned, I'm interested in the connection between the brain and the body. And this actually goes back to a quote from Darwin. So this is from Darwin's book, Expression of Emotion in Man and Animals. And he's talking about the work of the French physiologist Claude Bernard. And he says, Claude Bernard also repeatedly insists, and this deserves a special notice, that when the heart is affected, it reacts on the brain, and the state of the brain again reacts to the pneumogastric nerve on the heart, so that under any excitement, there'll be much mutual action and reaction between these, the two most important organs of the body. Now, this is a very interesting quote for a number of reasons. I want you to reflect for just a moment. What did we know in the 1850s about the brain? What did we know about the heart even? And specifically, how did they know that the brain and the heart were connected via this pneumogastric nerve, which we now call the vagus nerve? How? I have no idea. It has fascinated me my whole career. Now, what's interesting, so this is from Darwin's book, Expression of Emotion in Man and Animals. And It's depending upon the editions, approximately 600-page book. This is on approximately page 72. This is the basis of all of their work, not the conclusion. That to me is just completely mind-boggling, that they proposed that the connection between the brain and the heart was via the vagus nerve as the foundation for all their, what Darwin's future exposition of the importance of facial expressions, vocalizations, movement, and everything. His whole basis of emotion was based on this idea. Where they came from, where they got this idea from, a mystery. Two things, though. So Claude Bernard was a French physiologist, and the vagus nerve, the patron saint of wanderers, is Saint Julian. Yeah. The vagus nerve is called the wanderer. Claude Bernard was born in Saint Julian, France. I had no choice. This is what I had to study. Beyond my control. So with that in mind, I just want to give you a little bit of a background on some of the related autonomic physiology. So we talk about the autonomic nervous system in terms of these two divisions, sympathetic and parasympathetic. You can think of the sympathetic as sort of fight or flight, parasympathetic as rest and digest. And one of the things I'd like to emphasize, two things. One, many organs of the body are dually innervated. Most organs are dually innervated. The second thing is, the body doesn't know this division. The body doesn't know any of these divisions that we impose on it for our own pedagogical purposes. So it doesn't know the difference between the HPA axis and the autonomic nervous system or the immune system. All of these things are interconnected. And it's important to keep that in mind in trying to understand how these processes are working. The body does not know these distinctions. It's simply trying to mount a context-appropriate response to environmental challenges. And everything that the body does is in the service of that. And it's been hypothesized that the vagus nervous system is responsible for the psychophysiological balance of the organism. So the vagus nerve is thought to be the longest nerve in the body. It's the wanderer. And one of the interesting aspects of the vagus nerve is that approximately 80%, depending upon species, of the fibers are sensory fibers. So there are receptors on the vagus nerve for more or less anything that you can imagine. So cytokines, cholesterol, glucose. The vagus nerve transmits pain information, pressure information, all of this to the brain via the nucleus tractus solitarius. I don't have time to get into all the interesting physiology of the NTS, but there are vagal afferents as well. So some of you may know about vagus nerve stimulation, which can be done in a number of different locations, one including through the ear, through the auricular branch of the vagus nerve. I'll talk a little bit about some vagus nerve stimulation studies later. But there are all these vagal afferents that have been known for a long time. So the human dive reflex, stimulation of the ophthalmic branch of the trigeminal nerve, which produces bradycardia and oxygen consumption, reductions in oxygen consumption. And there is a reflex called the oculocardiac reflex that you may or may not know. The extraocular muscles also through this trigeminal nerve serve as vagal afferents. So in clinical medicine, ocular compression had been used to interrupt ventricular tachycardia. When your mom put a cold compress on your forehead when you were sick, she was stimulating the vagus nerve. Okay, so all of these vagal afferents feed interesting information to the brain, which is then processed. So this is a figure showing that there are actually two vagi. There's a left and a right. They have somewhat differential innervations, but it shows the output regions. The AMB is the nucleus ambiguus. The DMNX is the dorsal motor nucleus of the vagus. And they are the motor output sides of the vagi. And then it goes through the nucleus tractus solitarius all the way up to the prefrontal cortex. And I will illustrate that shortly. So over the years, we developed a model we call the neurovisceral integration model that suggests that there's a network of neural structures that can be indexed by heart rate variability, which is a measure of vagal function, which I'll describe here shortly. Higher heart rate variability is associated with greater prefrontal inhibitory tone. And a lack of inhibition leads to undifferentiated threat responses to environmental challenges. This last sentence is part of our thinking that we have formalized in our generalized unsafety theory of stress model, GUTS. And the idea here is that the default response is a sort of fight or flight response. That's actually a highly preserved adaptive response. So there was some discussion earlier today, a question about sort of the evolutionary basis of some of these things. So from an evolutionary perspective, this is an adaptive response. So sort of imagine walking along a path in the woods, you encounter something coiled on your path. Could be a harmless vine or a deadly snake. What's the adaptive response? Assume it's a snake. So from an evolutionary perspective, those organisms that had a strong fight or flight response in response to this potential snake lived to pass their genes on. So all of you sitting here, some of your ancestors had a strong fight or flight response. You know, the other outcome where on the off chance, so the point is that even if it's a harmless vine, the adaptive response is still to respond as if it's a snake. Because, you know, 9 times out of 10, it's a harmless vine, but that one time, you don't want that to be the time that you encounter the snake. So it's very highly preserved response across all organisms, and it's ultimately adaptive. Now, the neural structures that are involved are throughout the neuraxis involve cortical components, midbrain pons, medullary level. These are where the vagal outputs are. There's bidirectional communication throughout the neuraxis. And for our discussion, what's most interesting is that the output of the set of neural structures goes to the atrial node of the heart via the stellate ganglia and the vagus nerve. And you know, you all have seen ER or some other shows, and you've seen these traces going along. It's interesting. I've watched these shows, and you know, I've watched those monitors in these shows, and oftentimes the person is like, you know, dying or about to die, and their heart rate's 120 beats per minute, and it's like, no, it's not really accurate. So this is just sort of the nerd in me watching the monitors in the background of medical shows. But the timing between the heartbeats is what we're interested in with respect to heart rate variability. And you can see this variability manifest itself. The blue is respiratory mediated, relatively fast variability. The red indicates a sort of slower trend in variability, which is thought to be associated with baroreflex mediated blood pressure variations. And one can quantify heart rate variability in the time domain or at the top or in the frequency domain. Some common measures might be the standard deviation of the heart rate. One time domain measure that we use a lot is called root mean squared successive difference. It's from the econometrics literature, or engineering, is called first differencing. It's a way of essentially working as a high pass filter, so the slower trends are removed, so that that number reflects primarily the high frequency variability. In the frequency domain, one can use spectral analysis, and On your right side is one from an autoregressive spectral plot. And the vagal influences over the whole range. Sympathetic influences roll off at about 0.15 hertz or so. So that area in the yellow, the high frequency heart rate variability is due almost exclusively to vagal influences. And this is a rodent study from 2005 where they directly measured vagus nerve activity and this high frequency heart rate variability. And you can see that there's a very strong relationship between vagus nerve activity and this high frequency heart rate variability. Recently, Vaughan Macefield in Australia has been able to directly measure vagus nerve activity in awake humans and find sort of similar associations. So we're working with Vaughan to get his data to do the heart rate variability analysis on, and I took out those slides for the sake of time at this talk. So some years ago, we published a meta-analysis looking at heart rate variability and the neuroconcomitants of such heart rate variability. And in the yellow, we identified sort of three regions, two regions in the prefrontal cortex, the pregenial anterior cingulate, subgenial anterior cingulate, primarily on the right side, and the amygdala primarily on the left side. Now, this prefrontal amygdala circuit is thought to be sort of a stress circuit. So Richie has done some pioneering work looking at prefrontal amygdala relationships, and that those two regions of interest form a very interesting circuit that we have shown, and I will show you shortly, has been correlated with heart rate variability, connectivity of those two regions. So in this context, the brain regions that have been linked with heart rate variability are associated with important functions. This is a categorization from Amodio and Frith. The more dorsal regions associated with cognition, action monitoring, more rostral regions, emotion, social cognition, more ventral regions associated with reward, punishment, and outcome monitoring, and in particular, reports of safety. And that comes into play in some of the other data I'll present. But in the context of our generalized unsafety theory of stress, this brain region, this, excuse me, ventromedial prefrontal cortex brain region is particularly important. But what's also critical is that those brain regions are involved in autonomic regulation. Okay. So over the course of time, we've looked at heart rate variability and organism health at various different levels. With respect to physiological health, we've shown it's a predictor of mortality and morbidity. For example, a recent meta-analysis, we were able to show that heart rate variability predicted mortality. Interestingly enough for this audience, one of the samples that we used was the MIDAS data set, and we showed that heart rate variability predicted mortality in MIDAS. And also Whitehall. Those were two independent data studies that we had for our meta-analysis. I'm not going to talk about that today, for the most part. I will talk about sort of the index of emotional health and the attentional processes and cognitive health. And I'm going to do it in the context of what was alluded to earlier in Dr. Price's talk with respect to the issues with like the DSM and sort of the nosological categories, and the idea that taking a more RDoc perspective, a more dimensional perspective may be more appropriate. So in this situation, we wrote a paper a few years ago suggesting heart rate variability as a transdiagnostic marker of psychopathology. So this is just a few of the meta-analyses that we and others have published in support of this. So you can see heart rate variability, lower heart rate variability is associated with increased risk or increased prevalence of externalizing disorders such as alcohol dependence, internalizing disorders like anxiety and depression. ADHD is a little tricky, borderline, bulimia is an exception because of the nature of bulimia, which involves a vagal circuit, which is stimulated by vomiting, and also a lot of bulimics engage in excessive exercise. So exercise is associated with elevated levels of heart rate variability. And then we see depression in children, and then also with thought disorders with schizophrenia. So we propose that heart rate variability might be a transdiagnostic marker for psychopathology. This is just a little bit of the data on more physical health, but the top graph shows a relationship between heart rate variability and self-rated health. So individuals who rate their health as excellent have higher heart rate variability and in a dose-dependent manner compared to those individuals who rate their health as poor. And this association between heart rate variability and self-rated health was larger than the association between self-rated health and any other biomarker that we investigated, including measures of inflammation, glucose, blood pressure, cholesterol. I think those are the major ones we looked at in that particular study. So more recently, we were trying to identify a clinical cut point that physicians could use to identify people at elevated risk for a range of disorders. So this bottom figure just shows the association with blood pressure. So to orient you, this is a forest plot showing odds ratios for different discrete values of heart rate variability and its association distinguishing those who are normotensive from those who are hypertensive, that is, with a blood pressure of 140/90. And what you can see is that there's actually an inflection point and a value below which your risk for having hypertension is greatly increased. So over the range of risk factors that we looked at, we found odds ratios of 1.5 to 3.5 associated with one value, an arm SSD of 25 milliseconds plus or minus 4 milliseconds. If you had heart rate variability below that, you had elevated risk for a range of risk factors. I'll come back to that momentarily. This is another study at the top where we looked at a novel index of vagal neural immunomodulation proposed by the Israeli researcher Yuri Gidron. And again, this is in Midas and Whitehall. We were predicting mortality based on the ratio. So we had our measure of heart rate variability, our measure of vagal function over our measure of inflammation. And this is sort of consistent with Kevin Tracy's cholinergic anti-inflammatory pathway, where the vagal influences are inhibiting inflammation. So when your vagal activity is high relative to your inflammation, you had much greater survival compared to those individuals for which their inflammation was relatively high relative to their vagal function. So that's the top figure. When we just pulled out the heart rate variability, so this is from Whitehall 2, we found similar effect. And then if we used our cut point, on the right-hand side, you can see the survival associated with those above our cut point of 25 plus or minus 4. In this case, it ended up being 29. And those below that value had a much less survival. Relevant for the current talk about psychopathology, this is a paper we're submitting this week in about 10,000 individuals, where we showed that basically that same value of 25 milliseconds plus or minus 2 milliseconds in this case indicates elevated risk for depression based on the World Health Organization 5 depression measure. So think about it. You go into your doctor's office and you can get a 5-minute measure of heart rate variability that might identify individuals who have elevated risk, not only for pathophysiology, but for psychopathology. So we're kind of excited about the fact that there might be some ability to bring this into a clinical context. Okay, so in the context of this idea of linking heart rate variability to psychopathology, Caspi and Moffitt and colleagues proposed this P factor, so this general psychopathology factor in the structure of psychiatric disorders. The early work was done by Kruger, for example, where they had showed that this sort of higher order structure with internalizing and externalizing factors associated with these specific internalizing and externalizing disorders. This was expanded to include thought disorders. And what's interesting is that this general vulnerability factor was thought tended to overlap almost exactly with the thought disorder aspect of this. And what they note is that adults with higher levels of P fared less well on tests requiring attention, concentration, mental control, visual perceptual speed, visual motor coordination. Attesting to the ecological validity of these deficits, people who knew them well said that individuals with high levels of P experience cognitive problems in their everyday lives. So what I want to now show you is how heart rate variability is related to these cognitive issues that may form the basis of psychopathology. So we published a few papers, kind of review papers on this, but in the context of understanding the cognition and executive function associated with this, we relied on the Miyake and Friedman model, and they identified 3 components of executive function involving updating, shifting, and inhibition. And again, they found a higher order factor, a common executive function factor that was related primarily to inhibition. And this is what we think heart rate variability is indexing. And I'm going to show you some data in support of that now. So the first study we did, in the context of attention, there's the idea of intra-individual reaction time variability. So if your attention is focused, when you're doing a task, your reaction times will be very regular. But if there's any attentional lapses, there'll be random, more or less random long reaction times. And this can be captured in this first study we did was in a very primitive way with the standard deviation of the reaction times. So it was a modified flanker task, and we had individuals just press the button in the task appropriate congruent or incongruent trials. And you can see that heart rate variability was negatively correlated with reaction time variability for congruent, incongruent, and combined. And what's interesting about that sort of inverse relationship is that that highlights a phenomena called heterostasis. So you all know about homeostasis proposed by Cannon. Soon after Cannon proposed this idea, I'm not sure how soon, well, it was criticized more or less immediately, but I think it was 1959, R.C. Davis, one of the founders of the Society for Psychophysiological Research, published a paper highlighting that heterostasis was probably more common than homeostasis. And heterostasis involves variability in one system supporting stability in another. So the variability in heart rate variability supported stability in reaction time, in your attention, essentially. So this is just sort of a scatter plot of that. And I want to show this because you see that the distribution is not normal, and there are these long reaction times. And this has been modeled using what are called exponential Gaussian models, ex-Gaussian models. So the next study, we looked at heart rate variability and its relationship to these ex-Gaussian metrics. And again, we find that heart rate variability is associated with one component of the ex-Gaussian, the tau, which is a measure of those long reaction times. This is just a scatter plot. More recently, we used drift diffusion models to further explore this association. So again, here's the X-Gaussian showing the different parameters, and then the diffusion models. So my colleague at Ohio State, Roger Ratcliffe and Gail McCune, pioneered these drift diffusion models that have a number of different parameters. Drift rate is thought to be associated with this attentional control and inhibition. There's the boundary separation, and then there's non-decision time, which represents a motor component. And what we were able to show is that heart rate variability was associated with drift rate, particularly during those trials, those incongruent trials, when you needed to inhibit your attention. We also found, much to Dick Jennings' delight, that it was associated with the motor component, the non-decision time as well. So this is sort of laboratory studies suggesting that heart rate variability as a potential transdiagnostic marker of psychopathology may be associated with the cognitive inhibition that is thought to reflect the executive function, which is impaired in those people that have high P values. So we also showed this in more, let's say, ecologically valid inhibition tasks involving a rule shift cards task and a hailing sentence completion task. So these are just more sort of ecologically valid inhibitory tasks. We also found this in the think-no-think task developed by Michael Anderson in Cambridge, which is a measure of control over memory. I don't know if you're familiar with this think-no-think task. It's kind of like a cognitive no-go task, which of course is a prime measure of inhibition. The point is that you can actually not think certain thoughts. You can be cued to basically not remember. So you are trained up on a list of pairs of words. So for example, ordeal and roach. And then subsequently, you are given a cue, which can be either sort of the same prefix that you saw before, or that's called the same probe, or an independent probe. So a cue which is related to the category of the target word. So in this case, insect. But you can be told to not remember that association. And people can actually do it, which I think is kind of amazing. So your recall is lower when you're asked not to recall, so not to think, the think-no-think task. So what we were able to show was that those individuals with higher heart rate variability were better able to not think certain thoughts using either the independent probe or the same probe. Now, this has some interesting implications for psychopathology, because one of the issues in post-traumatic stress disorder, among other things, is intrusive thoughts. Now, bless you. One of the interesting additional aspects of this sort of cognitive control is that it's an issue in a lot of different pathologies. And the idea that, and I'll show you more detailed data momentarily, the idea that perseverative cognition, this ability, this worrying, rumination that is largely uncontrollable, is also a transdiagnostic marker for psychopathology. So those things are in fact related. We also, someone had mentioned fear conditioning, so I just threw in a couple of slides earlier to show that we actually find heart rate variability associated with inhibition in a conditioned fear paradigm. So we use the AX plus BX minus paradigm, where during the conditioning, you have a geometric symbol that's paired with either a shock or a non-shock. And what's interesting is the A-B condition, where you put the two symbols together. So does the person respond as if it's a safety signal or a threat signal? And then, of course, we look at the extinction as well. And what we find is that those individuals with lower heart rate variability were less able to exhibit fear extinction, and also were more likely to view that AB as a threat as opposed to a safety signal. So we published sort of a thought paper recently in Trends in Neuroscience, where we talk about these interactions in human fear conditioning, and just sort of the brain-heart connection via the vagus nerve, and how that is implicated in human fear conditioning, which may manifest itself in bradycardia. So moving right along, in addition to heart rate variability being this transdiagnostic marker, we proposed this sort of better safe than sorry idea as a common signature and, again, transdiagnostic marker of vulnerability to psychopathology. And one of the exemplars of the better safe than sorry strategy is this perseverative cognition. Worry, rumination, angry brooding, those are all aspects of perseverative cognition. So Emerson noted that life consists in what a person is thinking of all day. So what are you guys thinking about all day? It would be nice if you had some control over what you thought about all day. And I think the data I have showed you already, and some of the data I'll show you now suggests that those with higher heart rate variability may have better control over what they're thinking. And in the context of psychopathology, that's important. One of the things that you're trying to do with your patients is to get them to think about other things. So we propose what we call the perseverative cognition hypothesis. This is more in the context of physical health, how worry is associated with prolonged physiological activation. We did a meta-analysis showing that perseverative cognition was associated with things like decreased heart rate variability, elevated blood pressure, elevated cortisol, and like. So, human stress involves interactions, and then even when the person is not there, there's the perseverative cognition about it. And when this is extreme, it might lead to an anxiety disorder. It also can increase your health complaints. So we reported that not health-related worry, but just dispositional worry is associated with health complaints. So in an early study with patients with generalized anxiety disorder, we brought them into the laboratory, we had them relax, and we gave them a very detailed manipulation. We told them to worry as you normally do. That was the manipulation here. So two main effects, no interaction. The GAD patients had lower heart rate variability, but if you ask anyone to worry, their heart rate variability goes down. More recently, we were looking at the relationship between rumination and depression, and it's well known that one of the symptoms of depression is this rumination. And the question was, over this course of this prospective study over 2 years, was this association mediated by heart rate variability? This is just sort of the sample characteristics. And what you can see is that, bless you, is that controlling for sex, depression symptoms at time 0, rumination at time 0, was inversely correlated with heart rate variability at time 1, which was inversely correlated with depressive symptoms at time 2. And that heart rate variability, therefore, was mediating the relationship between rumination and depressive symptoms. Okay, so what I've tried to show you is sort of two potential transdiagnostic markers of psychopathology, one heart rate variability, one perceptual cognition, their association with different psychopathologies, and the fact that those two are in fact related to each other. So one of the things that happens when individuals come into your office as a clinical psychologist is that, they might come in and they say they're tense and nervous and can't relax. And the question you should be asking them isn't, where is your stress coming from, but why isn't it turned off in a relatively safe world? So some of us are, Michelle Krask and others, are viewing psychopathology and risk for psychopathology as a lack of recognition of safety signals. And this ties in with our guts model. So in this context, this is just one of the earlier papers on the guts model. The idea is that, it's not in response to specific stressors, but individuals who have ungeneralized safety have their default stress response on more or less all the time. So these people come into your office and they view the world as threatening. Yet it's relatively safe in modern times. So this is just how a model of how this might lead to prolonged stress responses and ultimately to psychopathology. So Ben Harrison's group published a very interesting paper looking at the ventromedial prefrontal cortex and processing of safety signals. They used sort of a conditioning paradigm. And what they found was that this same region that we found associated with heart rate variability was associated with recognition of safety signals. And in fact, the degree to which those safety signals were associated with positive affect was correlated with activity in this brain region associated with prefrontal cortex. So in a paper with Dan Fennell and Tony Buchanan, we looked at patients with damage to this medial prefrontal cortex region. So you can see, same region. They went through a paradigm involving an orthostasis task and a Trier social stress task. We had 18 patients with damage to that brain region, 12 patients with damage to another brain region, and 54 healthy controls. In the women, cortisol was elevated in those individuals that had damage to their medial prefrontal cortex. It didn't show up as much in the men, but for the men, their heart rates were elevated throughout the whole course. But what was interesting is that for both men and women, those with damage to that brain region, which is associated with safety, viewed the Trier social stress task as more threatening. So that brain region, which signals safety, when it's damaged, people perceive these things as more threatening. Now, threat is not always static. So, there's a concept of the defense cascade and the fact that threats can be dynamic, and they can be internal threats or external threats. I'll just show you one set of data from our colleagues in Germany, where the size of the geometric figure indicated the proximity of the threat or safe stimulus. And you could either sort of push your button to try to avoid it, or you had no choice. What was interesting was that as the threat became more imminent, activity in prefrontal cortex decreased and activity in the amygdala and paraqueductal gray increased. So there was a dynamic shift in cortical activity as a function of the proximity of the threat. Now, one of the things that we know is that individuals with psychopathology often have deficits in prefrontal activations. So it's possible that they're perceiving threat more or less continuously. So in another study, we looked at this proximity phenomena in the context of patients with panic disorder and agoraphobia. We identified 5 groups based on their heart rate response. So what we exposed these individuals to, this was a couple of hundred individuals, a multi-center trial in Germany on panic disorder. So we exposed them to a small kind of closet chamber that was locked from the outside. So it caused significant anxiety, you could say. We could see that there were some individuals that had higher vagal activity and those that had lower, and you could see this over the course of the 10-minute exposure to this enclosed chamber, using skin conductance as a putative measure of sympathetic activity. Again, we found the groups to differ. And in their exposure to this, there was a percentage of these individuals that basically said, let me out. So they asked to be released from the chamber. And then there was an even smaller number of individuals that actually experienced a panic attack in this chamber. So this is just sort of the heart rate response. And you can see that just prior to active escape, heart rate increased. And of course, prior to acute panic attack, it increased. But when we looked at the data more carefully, one of the things that we saw when we looked at RMSSD as a measure of vagal function was that there was an attempt, there was an increase in heart rate variability initially, an attempt to regulate their emotion. But as you can see, over the course of the 1 minute just before escape, heart rate variability went down. And in the 10 seconds just prior to escape, their skin conductance went up. Their sympathetic activity went up just prior to escape. But the interesting thing is that they tried to regulate. And you can see this also in the context of the panic attack as well. So 10 seconds prior to the panic attack is when the heart rate variability also changed as they were trying to regulate. And you can actually see this in the data of single individuals. So this is an individual 120 seconds prior to escape from the chamber. And you can see initially their heart rate fluctuates more widely, their heart rate variability goes up, but at about 60 seconds, there's just a rapid decline in their interbeat interval. That is a rapid increase in their heart rate. And by the end, their heart rate was up to 146 beats per minute as they were requesting escape. So you can actually see this attempt to compensate, and then it just doesn't work. Okay. So this vagal activation may be a kind of a resilience factor. And if you can increase this vagal activation and activation of these cortical regions, one might have some beneficial effects. So the next few slides are of a trial that I did with my colleague Mara Mather that was completed recently. And we brought people in and we recruited a bunch of individuals, young and old. The old sample got interrupted by COVID, so we were not able to get the full complement of participants, but in the end, we had over 150 participants, young and old. We sent them through a whole battery of things, but we had them do a biofeedback training, and this is for the increase group. So we had them try to increase their heart rate oscillations by paced breathing, and we identified individualized optimal breathing frequency, but it was more or less around 6 breaths per minute. And what we told them was meditation is known to have remarkable effects on emotional health outcomes. It can calm you down and reduce your stress and anxiety. Both the oscillation plus and the oscillation minus groups were given that same instruction. Here's where they differed. The oscillation plus group was told some meditative practices lead to large but smooth oscillations in heart rate called coherence. The oscillation minus group was told some meditative practices lead to a low and steady heart rate. Those were the differences in the two conditions. And then they were reinforced for producing the appropriate responses. You can see on the bottom that the heart rate variability in the 0.1 hertz, basically 6 breaths per minute range, was increased in the oscillation plus group, no difference in the oscillation minus group. So individuals did that for 5 weeks. 5 weeks of this training. They were brought into the lab, shown how to do this training, took home a device to aid in their biofeedback. They were asked to practice this starting for 20 minutes, ramping up to about 40 minutes a day. So one of the things that we found was that the connectivity in emotion and interoceptive regions increased as a function of this manipulation. It did not have a significant influence on other networks, so it was primarily emotion and interoceptive. We subsequently looked at the medial prefrontal cortex amygdala seed regions. We had previously published a paper showing that connectivity in those brain regions was associated with higher heart rate variability. And what we found as a result of the trial was that the oscillation plus group showed an increase in that connectivity post-training. We also published a meta-analysis, again, over 1,000 individuals across more than 20 laboratories across the globe. And we looked at relationship between cortical thickness and heart rate variability. And we found that the lateral orbital frontal cortex, some medial prefrontal cortex and insula regions were associated with higher heart rate variability, controlling for BMI, age, sex by age, and what research group. And this just graphically shows this. So our trial also looked at cortical thickness, and guess what? This 5 weeks led to increased cortical thickness in that same region that we'd found in our meta-analysis to be associated with heart rate variability. Okay. So breathing is one way in which you could increase your resilience potentially. For the sake of time, I might skip a couple of things here. Another way is transcutaneous vagus nerve stimulation. So we took a group of high worriers. We asked them to worry. We used a auricular vagus nerve stimulation or a sham condition. And we found that for spontaneous worry, as opposed to instructed worry, the transcutaneous vagus nerve stimulation was associated with decreased numbers of thought intrusions. And you could see this pre-worry. And the post-worry was kind of interesting as well, in that there was an initially larger number of intrusive thoughts in those that had received the vagus nerve stimulation, but they had a much more rapid decline after they had been asked to worry. So they were actually able to recover more quickly from the induced worry compared to those that had not received the vagus nerve stimulation. This is just the heart rate variability data associated with it. We also did a study where we used TCDS to inhibit the dorsolateral prefrontal cortex, again, in people who were high worriers. And we found that by basically inhibiting that activity, they showed greater sort of focus on the task but lower heart rate variability and greater cortisol responses. I'll skip this one. This is a recent paper from the same trial where we showed that this 5 weeks led to decreased beta amyloid levels. Again, just some of the details of the trial. I'll jump to the data. You can see that the oscillation plus group showed decreases in beta amyloid 42 as well as 40. So there are significant effects that our manipulation can have, our breathing manipulation. They are similar to effects that one might see with vagus nerve stimulation or transcranial brain stimulation. So you can stimulate from sort of both sides is the point, is that it's a circuit and you can intervene in different parts of the circuit. Now, just the last couple of slides here. In a study done with some colleagues in Italy, we took preschoolers, 4-year-olds, and we followed them for 4 years. And we looked at their exposure to early stressful events, and their heart rate variability changed over the course of the 4 years. And we recorded the number of stressful events that they were exposed to. And what's interesting here is that on the right-hand side, if they were not exposed to many stressful events, they were more or less okay, and it didn't matter what their heart rate variability trajectory looked like. But those that had been exposed to a high number of stressful events, if they had relatively high heart rate variability, they had better outcomes, similar to those that had not had many stressful life events. We found a similar effect when we looked at individuals much older in life who reported early childhood maltreatment, but the death of a spouse. And we found that if you look at the right-hand panel, those who had higher heart rate variability, but more childhood adversity, the dark line are the ones with a higher heart rate variability. Over the course of the months post-loss, they actually had less grief symptoms. So again, a potential resilience factor. Interestingly enough, we also looked at whether heart rate variability, I know again, in one of the earlier talks it was mentioned, are there any, I think Richie's question was there something pre-treatment that could predict the treatment outcomes? And in this case, we show that pre-treatment heart rate variability predicts dropout and residual symptoms. So those individuals that had higher heart rate variability pre-treatment were less likely to drop out and had fewer residual symptoms. And this was particularly true for those individuals who, their treatment dropout was due to motivational reasons. That is, they could not tolerate the therapy, as opposed to things like having moved away. And this is just sort of the statistics on that. We have unpublished data where we show a similar effect in response to pharmacological treatment. So Dr. Price, one of the things you might want to do is collect some heart rate variability to see if that predicts your ketamine response. I'm your man. And then, Chiara, in response to your talk, I added a couple of slides at the end on some of our sleep work with Sarah Mednick. So you raised so a couple of things. One, heart rate variability changes the function of phase of sleep. Heart rate variability is higher during stage 2 and slow wave sleep. And in this paper, we propose, based on work collected by Sarah, that there's a slow oscillation switch. So you were wondering about, the data where some types of reconsolidation or consolidation were enhanced and others were not. So we have an idea of why we find some effects on long-term memory and other effects on working memory, that we think that there's a switching going on throughout the night that's associated with the sleep spindles, actually, that are triggering this switch. And we can see this in the autonomic changes that are accompanying these brain changes. So we have this model that we've proposed, and we see the brain regions and the neuromodulators and the sleep mechanisms involved. So that came out in PNAS last year. So just to try to, the last few slides were just to try together the talks that you heard earlier. So I've run over by a couple minutes, but I want to end with a quote that's often been attributed to Darwin, but it's actually due to Megginson. He says, according to Darwin's Origin of Species, it is not the most intellectual of the species that survives. That's bad news for some of us in the room. It is not the strongest that survives. That's bad news for others of us. But the species that survives is the one that is best able to adapt and adjust to the changing environment in which it finds itself. And I'd like to propose that heart rate variability is a marker of that ability to adapt and adjust to the changing environment. And that if one can increase one's heart rate variability, again, I didn't have time to go into all the different types of ways in which that can be done, other than talking about vagus nerve stimulation, brain stimulation, and breathing. If one can do that, one might get better outcomes in a lot of different conditions, both pathophysiological and psychopathological. Thank you.
Thank you so much, Julian. That was a treasure trove of treats. So Danielle Carroll, a graduate student, and Sophie Woljan, postdoc, will lead us in questions.
Yeah, I know it was like a fire hose of data. I was just hoping there was something for everyone, a little bit of something for everyone.
Okay. Yeah, it was a really impressive fire hose, though.
Thank you.
A lot of really, really interesting work. So I have a question about, so you presented some work that's sort of looking at heart rate variability as sort of like almost a stable trait in people. And then you've also presented some work that's looking at it sort of as a mutable thing that you can actually change within a single trial. And so I think I'm interested in this more stable trait resting or heart rate variability and how that might actually change if you perturb the system in a more dramatic way, like say somebody with like a heart transplant or something like that. Would you find like different links between psychopathology and heart rate variability in subjects like this?
So there are a couple of components to your question. So one is whether we can think of heart rate variability as a trait measure or a state measure.
Mm-hmm.
So studies have been done using something called latent state trait analysis, among other things, and we've looked at it in some other ways. And we've also done both molecular and behavioral genetic studies. So I'll go from the reverse. So on the molecular genetic side, we've identified some SNPs that seem to be more or less generally across the general population. From a behavioral genetics perspective, we've identified sort of gene-specific variants associated with heart rate variability. From the latent state trait side, about 60% of the variance is more or less trait variants, but it is modifiable. So from the behavior genetics perspective, about 50% of the variance is heritable, which is what we often find in these cases, suggesting that it is mutable. And there are ways to modify it. Diet, exercise, breathing, all those types of things. And then at the acute level, increases in heart rate variability, as you saw in the panic patient, may index activation of those brain regions associated with emotion regulation. Again, we have a whole series of studies I didn't get to show you that sort of tie those in. So heart rate variability can be thought of as both a independent variable and a dependent variable.
Mhm.
And that confuses a lot of people sometimes. So the fact of the matter is that most measures that you take have some amount of trait variance and some amount of state variance. And if you know what they are and you can work with them, then you can interpret the findings appropriately.
Mhm.
Did I sort of answer your question?
Yeah, I guess to sort of ask the second part again. Yeah, so like what do you think if somebody were to have a heart transplant, do you think any sort of differences would occur in terms of their
Yes. So the heart transplant, of course, there's the denervation, but some data has suggested there's actually a little bit of a re-innervation, and then they start to get their heart rate variability back.
Oh, wow.
Yeah. So
That's fascinating.
yeah.
Oh, it's working. Okay. Yeah, thank you for that awesome talk. It was super cool. I was curious about the so at the end there, you gave this discussion of a study that was trying to manipulate heart rate variability with biofeedback. So I'm wondering, based on the current state of the evidence, what kind of reach do you think this biofeedback therapy is going to have as a therapeutic modality? Essentially, like, how excited should we be about the potential of heart rate variability biofeedback?
So, biofeedback in general goes back a long way. And Richie's mentors were some of the pioneers in biofeedback back at Harvard. So it's been around for a long time. There were some things that happened that sort of derailed biofeedback at a certain point in time. But I think it's coming back now, particularly in the context of heart rate variability biofeedback. I mean, you can go to a place like, some of these fancy spas like Canyon Ranch. They have a whole room for heart rate variability biofeedback. So at a certain sense, people are monetizing heart rate variability biofeedback, that's for sure. But I think, again, it was alluded to perhaps earlier or maybe during lunch, that the idea that we have to be very careful, because a lot of people, you can get heart rate variability numbers on your smartwatch. I mean, I've seen it on like television commercials for like insurance companies or something. And people talking about heart rate variability. And I kind of scratch my head, really? Do you have any idea what you're talking about? So I think there's potential good, but there's potential evil associated with it as well, because I think it's being monetized by people who may not fully understand what they're doing. So it's a double-edged sword at this point in time.
Okay. So I personally study social interaction. And so you're talking about this system that integrates the brain and the heart, vagus nerve, et cetera. And I'm interested in how that system might be expanded with the inclusion of another person. So how perhaps like long-term couples might, regulate each other's heart rate and what that looks like over a long period of time. Do you know anything about that?
Oh yeah, that's really interesting. So we published a paper with Tim Smith some years ago where we looked at married couples. And we had them interact in the laboratory about sort of a negative event, positive event, and then issue in their relationship. Okay. What we found was that the wife's heart rate variability increased during the discussion of the event in their relationship, and it was specifically associated with regulating the negative emotions of their husbands.
Great.
Yeah, those results were really kind of interesting. Stuff that you kind of knew, but it's interesting to have data just to show this. So, well, one of the interesting things is that, again, we've published a meta-analysis on this. Females, and I say females because it's not just humans, females tend to have higher heart rate variability than males. And it's thought that this is related to their emotion regulation, the need to regulate emotions in lots of social situations. So we find this increased higher heart rate variability in both, in rodents, primates, and humans at this point in time.
Very cool.
Sure. Yeah, I was wondering if you have any thoughts about the role of heart rate variability in music therapy and also communal practices of music, like singing in a group, or the evolution of groups creating musical practices together culturally.
So that's very interesting. As Richie alluded to, I am a musician, and my dissertation was on the physiological effects of listening to music. So we've been looking at this for a while. There are a couple of interesting aspects of this. In a music therapy context, it seems to be helpful. One of the things that we've shown, again, I think it's unpublished at this point in time, the student who was working on this disappeared. Those individuals that had higher heart rate variability were actually more sensitive to changes in musical parameters, which I think is very interesting. So they were actually sort of more sensitive to those environmental cues that were coming from the music. Now, with respect to the communal aspects, a couple of individuals, Torres Turrell has done a lot of work on sort of singing in groups and how that is associated with improved heart rate variability. And Elaine Chow has looked at this in musicians and their interactions with audiences and how that alters their physiology. So there are sort of group aspects of it, as well as individual aspects of the potential beneficial effects of music on things like heart rate variability. And I'll just mention, we just got what we think is a fundable score for a multi-center grant to put together a network of music researchers. So hopefully we'll have even more data coming out in the near future.
It's great. So let's open it up to the audience. I'll start. Julian, and this is a specific question about one particular data set. So you presented data on relations with beta amyloid. And I didn't quite get what the oscillation minus group is.
Yeah. So the oscillation minus group got the same sort of amount of feedback, but what they were, what their target was to keep their heart rate low and steady. Whereas the other group, the oscillation plus, was to increase their heart rate oscillation. So that's why we call it oscillation plus versus oscillation minus.
Okay, because it looked like in some of the data that the effect was carried as much by the oscillation minus group as it was by the oscillation plus group.
I know. Yeah. So that's one of the things that we're exploring in the new trial. So we're going to include another type of control condition besides the oscillation minus control condition that we used. You know, our initial thought was that we wanted to match these two conditions as closely as possible. And we thought same sort of biofeedback, but just with sort of a slightly different target. But those effects for the oscillation minus group suggested that we may need to look at a different control group to try to tease apart what is actually going on there. But you're right, Richie, you sharp eye.
I mean, that itself, though, is intriguing.
Yeah. So, you know, it would imply that if you're not doing good breathing, that, you know, you're going to potentially have some decline in or lack of clearance of the beta amyloid, which would be healthy. And, you know, historically, people don't breathe well. You know, we tend to breathe shallowly and from the upper chest areas. And I know that I just blanked on his name, who wrote the book Breathe. Yeah, exactly. So he argues that, you know, we need to learn how to breathe. So, yeah, thanks.
Thank you.
Hey, Julian,
Okay.
we have a couple questions, virtual questions back here. I'll just ask you. One is, can you comment on the use of heart rate variability monitoring devices, such as the Leaf monitor? And this must be a therapist, but do you recommend prescribing one of these devices prior to intensive psychotherapy for PTSD, for example?
So there are two questions there. So there are a host of different devices that are available. Polar Watch, Garmin, lots of different devices. There are apps on your smartphones that can get heart rate variability. We have validated some of these devices, like polar devices, against the EKG, and some of them are okay. Some of them are not so okay. There are new devices being produced even as we speak. And part of the issue is that their hardware and software tends to be proprietary. So we actually don't know what they're doing. So that's part of the problem with particular devices. One would have to do their due diligence to make sure that they got a device that's actually giving accurate information. Now, the second part of the question related to things like PTSD. So there's been large studies showing that individuals who had, so this is in a military context, pre-deployment heart rate variability predicted the risk for getting PTSD. I think that it has the potential to identify individuals who are at risk for developing, the whole range of psychopathology that we talked about, with PTSD in particular.
Here's a microphone.
Hi, I'm an undergrad here at UW in a class on sex differences in development. And I'm curious, you mentioned that heart rate variability is higher normally in females, but psychopathology during adolescence tends to be worse in females as well. And I'm curious if you see as well that variability in heart rate variability kind of increases during puberty when psychopathology emerges.
So that's a very good question. And my colleague, Julian Koenig, is sort of an expert on development of heart rate variability in psychopathology. And he's written a number of interesting papers on this topic. We've done some work together looking at this. In the context of the developmental issue in the sex difference, there may be critical periods. We sort of know about early critical periods, but there may be one around adolescence that may be particularly lead to particular vulnerability. And he's done a lot of work on that. One of the interesting things in this context, though, is more broadly, again, across species, we've shown that depressed females actually have higher heart rate variability than non-depressed females. And again, we think it's in the context of this attempt to regulate emotions. So we published a paper, numerous papers in humans, but one in particular in primates, where we showed that depressed monkeys, female depressed monkeys, had higher heart rate variability than non-depressed female monkeys. Now, why would this be across species? Again, we think it's the emotion regulation, because those depressed monkeys tend to be of lower social rank in these primate colonies. So that's what we think is going on. Now, the underlying physiology is also interesting. So the effects of acetylcholine, which is a primary cholinergic neurotransmitter, the effects of acetylcholine at the simul atrial node of the heart is moderated by estrogen. So, and again, some unpublished data, we have postmenopausal women with and without hormone replacement. And those postmenopausal women on hormone replacement actually had higher heart rate variability than those without. This is just sort of not depressed women. We recently, so the association between heart rate variability and heart rate differs between males and females. And again, I say males and females because we've found this in rodents also. So there's a stronger connection between heart rate and heart rate variability in females. So in humans, for example, for a given heart rate, say 80 beats per minute, the female will have higher heart rate variability than the male. And there are some structural differences that have been identified with respect to the intercardiac neural network involved. And females have fewer of these intercardiac neurons, but they may be more effective, which may be sort of the physiological basis for these findings that we have.
Hi, Julian.
Hi.
You mentioned the potential increases in heart rate variability or vagal tone through things like exercise or diet. What are your thoughts on long-term improvements, I guess at the risk of mixing physiological metaphors, like sort of a muscle where you're actively regulating, I guess using the female vagal advantage as an example, actively having to regulate emotions while in order to successfully navigate, say, a male space or to regulate your emotions in those contexts. Doing that on a frequent basis, do you suspect that that is working as a similar sort of manipulation or long-term improvement in vagal tone?
It's a little bit complicated, but so I indicated that the females tend to have higher heart rate variability than the males. African Americans have higher heart rate variability than European Americans, which would be contrary to what one would think, because you would think higher heart rate variability, better health outcomes. So we've identified a pattern we call the cardiovascular conundrum, where the African Americans have higher heart rate variability, which should be cardioprotective, but they have higher blood pressure and higher total peripheral resistance in particular. And in several studies, we've shown that in one prospective study where we were predicting total peripheral resistance 6 years into the future, higher heart rate variability was associated with lower total peripheral resistance in whites, but not in blacks. Now, we think that this higher heart rate variability represents this emotion regulation. And we have some evidence from this from a large study in Brazil, where in Brazil, there are sort of, it's a very ethnically mixed group, and there are at least sort of 3 phenotypically defined ethnicities, the English translations of which are, black, brown, and white. And we found that the blacks had higher heart rate variability than the browns, than the whites, and blacks were exposed to greater discrimination, again, in a dose-dependent manner. Blacks also had higher rates of hypertension. But when you controlled the heart rate variability difference for their exposure to discrimination, the heart rate variability difference was attenuated. So we think it's that they're trying to regulate their emotions. Now, one of the arguments we get for the relative risk of health disparities in the US is that it's genetics, but or diet. So there's an old theory that should have been thrown out long ago, but is not, about the Middle Passage, and that the blacks that came over the Middle Passage had to retain salt, had to retain water, and therefore more salt sensitive. All that is nonsense. There's no real evidence for that at all, but yet people still invoke that theory. So one of the things that we did was to show that this conundrum pattern was actually a response to unfair treatment. Was we, with Christina Ottaviani in Rome, we had a sample of lesbian, gay, and bisexuals, and they showed the cardiovascular conundrum pattern too. So there's no way that they have the same genetics as the blacks, and there's no way that they're eating the same food as the inner city blacks. It's their response to unfair treatment. So that's what we think, that this higher heart rate variability is an attempt to regulate, this chronic stress. And it ultimately leads to a breakdown of these compensatory mechanisms, because the fact that their higher heart rate variability should be associated with lower blood pressure, and it's not, suggests that that mechanism is somehow corrupted. So in a whole other series of studies, we have shown that it's related to the vascular branch of the baroreflex, that the blacks have less effective vascular branch, and that this is associated with lower nitric oxide availability, which on the other hand is associated with increased inflammation in Blacks, which is the way that because there's no direct vagal innervation of the vasculature, it goes to this cholinergic anti-inflammatory pathway. That's our working model at this point in time.
Oh, okay. Thank you for that wonderful presentation. My name is Hadley Rarick. I'm another trainee here with the training program in motion research. So your work has framed, HRV as a mechanism of emotion flexibility. And this is why populations with mood disorders tend to exhibit, lower HRV. So my question is for you, how would you conceptually or empirically differentiate an adaptive flexible response to environmental threat from an unstable or histrionic pattern of reactivity to daily stressors?
That's a good question. So on the basis of just the physiology, it's going to be tricky. So you have to, and the way that we do this, and Richie alluded to it, is we look at it in a broader context. So sort of in terms of a pattern of response, both physiological and psychological. So in response to the previous question, when we look at the heart rate variability in isolation, we would assume that the Blacks and the women would have better mental and physical health. But when we look at it in a broader context, we see, oh, okay, there are some other physiological and psychological consequences of having that heart rate variability that doesn't make it adaptive. So just looking at the heart rate variability itself can't tell. You really need to look at it in a multivariate way.
Sure. I'm thinking of an example where Richie and I were looking at a pattern of daily emotion responses on, a negative affect to positive affect scale.
Mm-hmm.
And our lab was trying to determine, would we conceptualize this pattern of responses as somebody being overreactive or unstable in their emotion characterization from moment to moment, or are they actually having an adaptive response, a flexible response?
So one of the things that we have found is the way that we think about it is it context appropriate?
Mm-hmm.
So there may be a lot of variability, but if it's not really sort of matched to the environmental demand, so there are situations in which your heart rate variability should decrease, for example. And if it doesn't decrease in those situations, then having that higher heart rate variability is probably not adaptive.
Mm-hmm.
So again, it's kind of a matching of the physiological response to the environmental demand, let's say.
Mm-hmm.
So you would want to look at it over time then. Just a single snapshot, you might not be able to tease apart whether that's an adaptive response or not.
Thank you.
Thank you so much. This is really great work. I've been reading it since undergrad, which feels like a lifetime ago. So
you're trying to make me feel old is what you're saying.
I'm trying to see how, what I can do to your HRV. So you mentioned in your biofeedback slides talking about individualizing optimal breath patterns.
Yeah.
How do you do that? It's extremely pertinent to my work right now. And what are the, how variable are the individual differences in breathing, and how do you measure what's really best? And I don't know of any other work, though I haven't looked that far, that's done that. And I'm really interested in it.
So Paul Lehrer is one of the collaborators on our on that project, and he has an individualized way. So by looking at the actual heart rate oscillations, have people breathing at somewhat different breathing frequencies, we identify for each individual the breathing frequency for which is the highest. It's in the more or less 6 breaths per minute range, plus or minus a half a breath or a breath difference. So yesterday we had a call about another project that we're working on where we're not going to be able to do the individualized. And we agreed that, well, we'll just have everybody do it at 6 breaths per minute. If you have the opportunity to do it on an individualized basis, there's a little bit of added value to that, but it's not, doesn't account for the whole effect.
Thank you very much.
Hello, thank you for your talk. I have kind of a two-parter, I guess. And the first part is, can you, I guess, speak a little bit on what the heart rate and heart rate variability would do during like an active, like physical pain sensation? Yeah, I'll pause there.
Okay, so again, with my colleague Julian Koenig, we did a whole series of studies on heart rate variability and pain. So there are 2 aspects of this. One is that individuals that have higher heart rate variability tend to report less sensitivity to pain, you could say. So we've looked at this both in the context of individuals with chronic pain, they tend to have low heart rate variability, and also in the context of acute pain. So there's a whole series of studies on that you can look at. In a recent study, we found that this is with Yuri Gidron and some colleagues in Italy. These were patients undergoing brain surgery. And we found that those individuals that had higher heart rate variability pre-surgery needed less pain medication after, which we thought was kind of interesting.
That is really interesting. And so I guess that's kind of related to my second part. And I know you talked a little bit about orbital frontal cortex stimulation. Do you have a hypothesis as to what would happen with dorsolateral prefrontal cortex stimulation?
So we've done the dorsolateral prefrontal stimulation Voigt and Helen Mayberg and some others have published a very interesting paper on they sort of did a biochemical dissection of the connectivity of these different brain regions. So these sort of more dorsal regions have a more in a certain sense, direct connection to the ventral regions than even maybe the adjacent rostral region, which was kind of interesting. And that sort of fits some of our data in that, the connectivity, so the effect on the heart is primarily going to be via these ventral medial regions, but that you can get access to it through that more dorsal region because of that pattern of connectivity. And you might, like, again, it's kind of curious because I guess the morphology or the phenotype, I'm not sure how exactly they characterized it, of the brain structures was more similar for the dorsal and this ventral medial than for some of these adjacent regions. So in one of our papers, one of our empirical papers on cortical thickness and heart rate variability, we detail that, the structural connections, because we found an effect on the more dorsal regions in that particular study.
Cool, thank you. I know Martin Arns has a paper about kind of the frontal vagal pathway and how that may mediate that cardiovascular effect, and that's something that I'm researching. So I just was interested in your, I guess, perspective on it. So thank you.
Cool. I'm curious to hear what you're working on.
So earlier on in the talk, when you were talking about the P factor, one of the things that reminded me of, is that related to the idea of the biological sensitivity to context?
That's a good question. I'm not exactly sure how Caspi and Moffat have characterized the P factor, but I could say that heart rate variability might be related to that, but I'm not sure if, I don't want to speak for those guys. They may think that or not, but I'm not exactly sure.
Thank you for this fascinating talk. I think I was particularly interested in seeing the transdiagnostic aspect of heart rate variability and how it presented across different contexts. So I think more at a broader level, in psychopathology, I'm interested on your thoughts, whether you think it's indeed risk factor or kind of biomarker mechanistic index, or whether it's more an outcome or a consequence of the clinical presentation.
That's a really great question. And I think there's evidence for both. That is to say, there was a paper, for example, by Vyryandakova using the Whitehall data, where she showed that heart rate variability predicted depression in the Whitehall data, like 10 years into the future, something like that. So lower heart rate variability preceded clinical symptoms of depression. So it may serve that that role. It also, John Allen and others have shown that it's a sensitive marker for treatment outcomes, potentially. So it can track potentially when people are getting better. So I think it can sort of function on both sides.
Thank you.
Julian, I have another question. So you, one of the early questions was about state and trait issues, and you nicely, I think, summarized the evidence in indicating that it really is both state and trait. A little later in response to the questions, you talked about the possibility that the failure to modify heart rate variability in response to specific contexts may be an important signal. And so this is a two-part question. One is, in the cases that you mentioned where, for example, depressed women have higher heart rate variability and Black African Americans have higher heart rate variability, at least in certain contexts. I assume, first of all, those are trait measures. And the question is, are any of those cases associated with the failure to modulate heart rate variability in a context appropriate way?
So that's a very good question. So one of the things that my colleague Dwayne Williams has been looking at are sort of within group variations in heart rate variability and how it's related to different types of factors. For example, exposure to discrimination and things like rumination. So sort of within group, greater rumination in Blacks is associated with lower heart rate variability to an extent. So, but one of the things that he has found and his
But that's still a trait measure.
yes, but I'm getting to it.
Okay.
So that I don't know if he's published this yet. What he found was that the resting heart rate variability was associated with the phasic response to this unfair treatment or these reports of discrimination, such that those that had higher heart rate variability as a trait, as they were exposed to unfair treatment, their heart rate variability went up phasically. But those who had low heart rate variability as a trait, when they were exposed, their heart rate variability went down. So again, and we've shown in other studies sort of this relationship between the trait and the state. So if you have higher trait, you might view this challenge as a or this situation as a challenge, and therefore mount a successful regulation response. Whereas if you view it as a threat, you might show this decrease in heart rate variability, which is
But
associated with stress responses.
Is there a relation between trait levels, baseline levels of heart rate variability, and the absolute magnitude of heart rate variability change in response to challenge?
Yes.
There is.
Yeah.
So higher heart rate variability is associated with a larger change.
Yeah.
Okay, so they're not really orthogonal.
That's right. Yeah, exactly. No, they're not orthogonal at all. Yeah.
Okay, thank you.
Yeah. And we've shown this not only in, for example, the example I gave you, but also in the context of patients with generalized anxiety disorder, for example. In the paper we published, I can't remember exactly when, we had the patients with generalized anxiety disorder, matched controls, and the magnitude of their phasic cardiac response, including their heart rate variability, was a function of their resting heart rate variability, such that the controls had a larger magnitude change compared to the patients who had lower resting heart rate variability. So we find it in a number of different contexts.
Yeah, all this discussion that we've had about the experience of discrimination and heart rate variability made me think back to our earlier discussion of the oscillation plus and minus groups in your study. And I was wondering if you think there's a possibility that the benefit that people might get from either of these groups could be broken down by experience of discrimination. Like, for example, people experience more discrimination, might they benefit from the oscillation minus group, or potentially the opposite way?
That's a very good question, and we just got a grant to look at it.
Right.
So I'll let you know in a couple of years.
You have time for maybe one more question. Any other questions?
Ned, you've been remarkably silent. Right.
I'm curious, thank you for the talk. It was incredible. I really appreciated that you mentioned sleep, and I'm curious. So I've read that sleep apnea can really interfere with a lot of the cognitive benefits of sleep. And I'm curious how sleep apnea might also disrupt HRV and just general heart rate during sleep.
Yeah, so sleep apnea is associated with decreased heart rate variability. Yeah, absolutely. It's really disruptive.
Thank you.
Well, thank you very much. Let's give Julian a round of applause. So,
Thank you.
I was actually looking at my Apple Watch as you were talking, trying to figure out how accurate that was in relation to my heart rate variability. Want to remind everybody that we've got a poster session coming up, and we've had spectacular talks today, which have been a little formal, but this is going to be a lot less formal. And so I want to invite everybody to come and to hang out, have a good time. There's stuff to drink and there's food to eat, and this is an important part of the meeting. So I expect to see everybody there, and we're going to have a good time. So thank you. And also tomorrow morning, we're starting at 8:30, I think. And also in the back, Amber, hey, Amber, wave your hand, will you? I want to recognize Amber Sebastian, who has not only has she been an incredible organizer of this meeting and has planned it out all the way along, but as you can imagine, she's put up with myself and Richie. And so that is no easy task, and she's done it with grace and a lot of emotion regulation and probably a high heart rate variability as well. So again, thanks again for the talk, Julian, and we'll see you outside at the poster session.
Thank you so much for your great question. So what are you guys working on?
So I'm working on social analysis, specifically like how we measure opposition. So across a bunch of different heart rate variability. I've never studied it yet, but I'm planning on bringing it to this place.
Oh, let me know what I need to help.
Yeah. I've been calling you in the class. So
Oh, yeah.
yeah.
Okay.
Yeah. So I
what I'm really interested in is how all of these different inputs to conversations, like being eye gaze, heart rate variability, and the way you're talking things together, like how these all the different like together build sort of like a model for how we connect to things as well.
That's pretty. Yeah. What do you work?
I'm in it's not what was on the side, so psychopathology and all this stuff. Markers of the body, right?
That's really.
Also.
So that's one of the things that we're going to be looking at in this new trial. And we posted a meta-analysis a couple of years ago showing the heart rate variability is negatively correlated with the full range of pro-inflammatory cytokines. So that whole cholinergic and inflammatory pathway, we're really looking at on the material.
An interesting thing that I noticed is that in my studies with it's been a lot of things are kind of I see that are how these are being it's the insula, right?